Most bacteria have a rigid cell wall that surrounds the plasma membrane. So again, we have our plasma membrane, and then we have a thick cell wall outside of that plasma membrane. The reason why they have these is because they help protect their cell shape and help protect them from attack from other cells, or for example, like the immune system. So a lot of protection functions. One of the things that it protects it from is lysis. So lysis means to break open, and this occurs in hypotonic. This can occur in hypotonic conditions. So if you have a hypotonic solution and you have a cell, hypotonic environments are high in water, so higher than the cell, so water will tend to flow into the cell. And if water flows into the cell, then the cell can eventually burst. But if that cell is surrounded by a cell wall, just like in plants, the cell wall provides a returning pressure. And so as water flows in, the cell wall pushes back on that swelling cell membrane, and so it will push that excess water out. So the cell wall helps prevent that. This is critical for bacteria because many of them live in environments that are water-based, so like a pond, for example, and they don't have control over their external environment. So we have kidneys to help maintain salt balance for our cells, but they do not. Their cells are very vulnerable to the environment, and the environment overall tends to be less salty than a cell, so their protection is against hypotonic environments. The cell wall of most bacteria is made out of peptidoglycan. And so this is a material that's a complex of proteins and sugars. That's where the name peptidoglycan comes from. And it's a specific combination of proteins and sugars. So they all have peptidoglycan in their cell wall, but how that cell wall is put together with peptidoglycan differs a lot, and it differs specifically into two distinct groups. And so many bacteria are classified as either gram-positive or gram-negative, and again, it depends on how that peptidoglycan layer is present in the cell wall. It's present in both of them, but in a different way. So the name gram-positive and gram-negative comes from something called the Gram stain. Like a lot of stuff historic in science, it's named for the scientist who invented it, who was named Gram. And it's basically just a sequence of chemicals and two different dyes. One is a purple dye, and one is, it's called red. The name of the dye is, but it actually kind of leaves a pinkish color when you look at it under the microscope. So what happens is, based on how exactly the cell wall is put together, it will either leave a purple stain in the cell wall or it will leave a pink color in the cell wall. And so that's how you distinguish them. You apply the Gram stain. So you do this whole sequence of staining, and it's a very, multiple steps to it. And at the end of those steps, gram-positive bacteria will appear purple and gram-negative bacteria will appear pink. So the reason why this is that the gram-positive bacteria have a really thick peptidoglycan layer. And so that thick peptidoglycan layer will hold on to the purple dye, and they will appear purple. Lots of examples of gram-positive bacteria, Staphylococcus aureus, Streptococcus pneumoniae, or Staphylococcus pneumoniae are both examples of disease-causing ones. Generally, the gram-positive bacteria, unless they're resistant, are vulnerable to the penicillin class drugs. So penicillin was our first and probably most famous antibiotic. Its relatives include things like ampicillin or amoxicillin. Any of them that have that ending are related to it. And the reason why they work on gram-positive bacteria is because they interfere with the production of peptidoglycan. And so because they have such a thick layer of peptidoglycan production, if you slow that down, it makes it hard for them to reproduce, right? So the antibiotic blocks reproduction of the cells. For these gram-positive ones, they basically can't make enough peptidoglycan to make a new cell. Gram-negative bacteria have a thinner peptidoglycan layer. So it's still there, but it's much thinner. And then on the outside of that peptidoglycan layer, there's an additional layer of lipopolysaccharides. So lipo is referencing fat and polysaccharides is sugars. So it's a mix of lipids and sugars. And so because they have a thinner peptidoglycan layer, the purple dye that is added doesn't stick. And so the second dye, the pink dye, called safranin red, but it actually makes it pink, will stain it instead. Okay, so the purple stays in the thick peptidoglycan layer. It doesn't stay in the thin peptidoglycan layer, so it takes on this pink color. An example is E. coli. These bacteria are more resistant to penicillin just naturally. So if somebody has an infection with one of these, a gram-negative bacteria, it has to use different antibiotics than the penicillin family because they don't have that thick peptidoglycan layer, so blocking peptidoglycan production just doesn't affect them as much. And this is why knowing which type of bacteria is causing an infection can be helpful in knowing the treatment to use. So this is what these differences look like. So remember, gram-positive is going to have a purple color when you look under the microscope, and gram-negative is going to have a kind of pinky color. Take a moment, too, to just review cell shape. So is this, what cell shape do these have? This is a little bit tricky because they kind of look a little bit round, but a little bit rod-shaped. So this would be a hard call at this level of resolution. I might actually call them cosi because I see some, especially when they're by themselves, some distinctly round ones. But you could also, without any further information or better resolution, these might be bacilli. These ones are nice because they're really clearly bacilli. So we can see that distinct rod shape. Okay, so that's just a little review of shape. But let's take a look now at their cell walls. So the gram-positive, this part here is that thick peptidoglycan layer. This is the membrane. These are various other molecules that might be embedded into that, like cell surface type sugars, for example, that might be sticking out of it. This is a gram-negative cell. So here's the cell membrane underneath here. Here's this little teeny thin layer of peptidoglycan. So again, look at that scale of difference. This is a huge thick layer of peptidoglycan. This is a really thin layer. And then on top of that thin layer of peptidoglycan is this complex of lipopolysaccharides. So you've got these lipid components with a sugar attached to them, making up this additional layer. And again, this difference in peptidoglycan is what's going to get you this difference in color, because the thin peptidoglycan and the lipopolysaccharide layer will cause the purple to not stay. And so this has medical relevance as far as knowing the difference between gram-positive and gram-negative. It affects treatment. And also, in many cases, it reflects some degree of evolutionary relationships. So especially the gram-negative bacteria are related to each other. And so that's why this classification is useful to us. Okay, as a last note here about specialized things that can happen on the outside layering of the cell. So we've talked about like the cell wall, the glycocalyx. Some bacteria have the ability to sort of modify the outside of the cell even more, in particular for surviving unfavorable conditions. So there's two examples here. Again, not all bacteria can do this or do either of these. Only certain types of bacteria can do either one. So the first example here is the formation of aconides. Aconides are special cells, and this is an example of one. This is the regular cell. And the Aconites has formed. It's bigger. It has a really thick cell wall, and it's filled with food on the inside. So as part of its like life cycle, these bacteria live in ponds, are photosynthetic cyanobacteria. And so in many cases, as winter approaches, the temperature changes, the food and light availability changes, they will be triggered to form the Aconites. They pack up with food and they go into a dormant state. This dormant state has reduced metabolism, but they still do some metabolism. So that's why they have to have the food. The food is helping them survive that small amount of metabolism. And then once favorable conditions return, so usually in the spring, then the Aconites will return to its regular cellular state. And so this can happen, again, on sort of a seasonal cycle, though it can also just be something bad happened, like a pond dried up or something like that. And so these Aconites have been known to persist for like 50 years even in some cases. So we do have this seasonal cycle to it, which makes sense for where these organisms live, but they can even last longer than that. Endospores are even more durable than Aconites. So again, some types of bacteria do this. What they do is they form inside of the cell. So this is a cell. Whoops, sorry, I kind of missed the, so let me do that. So let me use a different color here. So this is the cell membrane on the outside here, and this is the endospore forming. So it actually forms inside of the cell, and it's triggered by, again, sort of unfavorable conditions, environmental stress, not enough food, wrong growth conditions. This cell basically realizes that it's going to die. And if it realizes that it's going to die, it's going to start forming this endospore inside of itself. The endospore contains the genetic material and a super tough protein coat that's heat resistant. So Aconites are cold resistant and low food resistant, but they're not heat resistant. What's really unique about the endospore is that it's heat, cold, temperature, or I already tested temperature, salt. It's resistant to a lot of different stuff, and it is non-metabolic. So it doesn't store food the way an Aconites does. It's just not going to carry out any metabolism. So what's going to happen is this cell is going to die, and it releases the endospore. And then that endospore just kind of hangs around in the environment. And it can just hang around for a really, really long time, like hundreds of years, because it's not metabolic. And because it's resistant to all of these different extreme conditions. And then at some point, if that endospore gets back into favorable conditions for that cell type, it's going to be able to germinate and grow into a new metabolic cell. So some examples of bacteria that do this, and some of this may ring a bell for you: Bacillus anthracis, Clostridium botulinum, Clostridium tetani. So Clostridium tetani is the cause of tetanus. Clostridium botulinum is a source of botulism poison, or botulism, which is a form of food poisoning. Bacillus anthracis is the source of the anthrax toxin. So these are bacteria that can be dangerous. Now, for the most part, we're not exposed to them because all three of them are soil dwelling, and specifically deep soil dwelling. Deep soil as in no oxygen. So they live deep in the soil where there's very little oxygen penetration, and so they actually won't grow in conditions where there's oxygen. So what happens is you have like deep soil down here. If one of the bacteria get close to the surface up here, they die because they can't handle the oxygen, but you end up with endospores that are hanging out in this surface. Now, as long as it's the endospore, it's not going to hurt anything because the endospore is not able to germinate in oxygen environments. So like if it's like in your, on your peach that you ate, or on a piece of lettuce that you ate, it's not really going to matter because the endospore is going to be in oxygen. It's not making the active cell, and it's going to go into your gut, and it's just going to get broken down by the acid of your gut and never germinate into a live cell. However, these can become problems if they enter a low oxygen environment. So for example, if they get into the bloodstream and this or into your tissue, so like if you step on a nail that has the endospore on it, and that nail punctures deep into your tissue, it penetrates into an area that might have low oxygen. And then the endospore can germinate and initiate a tetanus infection inside of you. In the case of Clostridium botulinum, it can get introduced into a low oxygen environment by being, for example, inside of a can. So canned food creates a low oxygen environment. That's why it's preserving the food, is because regular bacteria can't break it down because they can't grow without oxygen. But Clostridium can grow without oxygen. And so if there was an endospore and it gets into the canned food, then there's the potential for that bacteria to germinate and to produce the botulin toxin, which causes botulism. Now, normally that's prevented because a lot of canned food is acidic. And so, as I mentioned with the stomach, that's going to prevent germination of the bacteria. So like canned tomatoes are acidic. Even if there's an endospore inside of that can, it can't germinate. This can become a problem if the food wasn't acidic enough, or if they got into something. So the most common, I'm trying to get to the most common type of botulism poisoning, which is actually in what's called prison hooch, or when people try and make alcohol, which you do in low oxygen conditions, but they're using something like potato peels, which introduce the endospore and is not acidic enough to prevent the production. It also happens in people who are IV drug users. So the endospore gets introduced from dirty needles, and then it kind of like with the tetanus, it's getting injected deep into tissues and can germinate there. So it's actually not very common in canned food is what I want to tell you. So I've told you the scary thing that it can be in canned food, but the cases of botulism poisoning from canned food is extremely, extremely rare. You really don't see very many of those. So in the medical setting, understanding endospores is really important because, again, if it gets introduced into deep tissue, then these bacteria can grow. And so in order to prevent endospores from being on surgical materials, or when we say a clean needle, for example, we have to use special treatments. So we use pressure and heat to eliminate endospores. Heat alone is not enough. And the pressure and heat is provided by a piece of equipment called an autoclave. So an autoclave is a special piece of equipment that helps to ensure that not just bacteria are gone, which you can do with just boiling water, but that bacteria and endospores are gone, and that requires pressure and heat. Hence the use of autoclaves, not just boiling water to sterilize medical equipment.